everybody. Great to be here. Great to see everybody. It's been a couple of years since we've been able to visit in person, and so really this is the first event I've done in a couple of years. So wonderful to be back out. Usually I'm in the lab up in Oregon, but try to get down here a few times a year, visit with scientists, see what you're working on, and really do educational events like this to talk about different technologies. So a few things for today. I thought we'll give a brief introduction to what flow cytometry is and how it works, and then spend some time looking at how we prepare our samples for flow, some different things to consider when getting your samples ready to put on the instrument, and then look at a whole range of different possibilities, different things you can do with flow. And at that point, I'll kind of open it up to you, and hopefully we can talk about what you're working on, and maybe see if we can troubleshoot what you're working on or discuss interesting findings you have. And definitely, as Serena said, questions are welcome, discussions welcome. So enjoy the lunch, and we'll go ahead and get started. As I mentioned, we built in, maybe 30 minutes of presentation at the max, but really having the goal to have the bulk of our time spent discussion about what you're working on and maybe how flow can help reach your goals. So I guess who has done flow cytometry before? So 3.5 maybe. I saw some. 3.75. Great. So we're about halfway there. So we'll spend a little time with the theory of how it works and why we would do this as opposed to microscopy as a single cell analysis platform. And then definitely the experts in the room weigh in if I'm off base with anything. So good to have experts here also. With flow, generally what we're doing is combining 3 different systems in one box. So 3 things are required for flow. One is the fluidic system that carries our cells from whatever reservoir they're in, usually a tube or a 96-well plate, carries our cells through an interrogation point, ideally 1 cell at a time, so that we can analyze 1 cell at a time. Those cells are passing through an optic system that's shining light on those cells. And because our cells are labeled with different colors of fluorescence dyes like RPE or FITSI, various dyes that show up different colors, when we shine light on those cells, different colors of light are given off. And then using a detection system, we can detect the light given off of every individual cell across the spectrum of possible colors. And then finally, an electronic system takes those pulses of light given off, tiny pulses of light given off of every cell, amplifies them, and then makes sense of it all in a way that makes sense to us in terms of data. And so what this looks like when they're all put together, I guess first, the way that our cells are focused in the fluidic system generally relies on a system called hydrodynamic focusing. This means using fluid to focus our cells, to get them into a narrow stream where they go from a wide sample with lots of cells moving very slowly to a very narrow stream that's moving very rapidly through the laser. Often this can be 5,000 cells per second, 30,000 cells per second that are passing through our interrogation point. But it's this squeezing that makes our cells go through very quickly. And so we can analyze, 10,000 cells or 1,000,000 cells or 10,000 exosomes or whatever you are studying very quickly. The instrument that you have on site has what we call sheath fluid. That is the saline solution that does this squeezing of our sample, but it also has a second focusing component called acoustic focusing. So it actually uses sound waves that are tuned to a certain frequency that pushes our cells. The actual sound, ultrasonic sound waves push our cells physically into this line, and that allows the cells in the instrument that you have to go substantially faster than any other instrument can. So essentially, you can get results a lot faster than other instruments are able to. So what we do, as our cells are passing through a flow cell, we have one or multiple lasers. On this campus, you have 4 lasers available that shine through this interrogation point, and the light given off is measured in different ways. One of the ways we measure that light is by looking at what's called forward scatter. This is essentially a measurement of, you could consider it the shadow of a cell. Like if you tossed a basketball across the court, you could see a shadow on the ground. It gives you an idea of how large that basketball is. It's kind of the same idea. So forward scatter is roughly the shadow or the size of your cells. We can also look at light that bounces off at 90 degrees, and this can give us side scatter, which is light that's bounced off at 90 degrees, and this gives us an idea of the complexity inside a cell. So if there are lots of granules, lots of lysosomes, or various sturdy structures inside a cell, light will bounce off and show up as side scatter or complexity. And the combination of these two things alone tells us a lot about what we're analyzing. But really, when we start to label our samples with fluorescent dyes, we can look at in different channels using kind of like how a prism separates light into its different components to a rainbow of colors. We use similar mirrors that split light into its different colors, and we can analyze how much of each color is given off from each cell. And so this is all processed and shows up in our instrument as forward scatter versus side scatter. Or we can look at how bright a cell is in a given color versus another color. And in the case of the Attune on campus, we can study 14 different colors, 14 different markers per cell. So like I think in the organ on a chip model, you might be looking at cells that express 1 or 2 given markers, and that identifies them as a fibroblasts or a myocyte or something like that. So we could have, for example, a fibroblast marker here, a myocyte marker here, or a neuron marker here, and we can identify different cell types based on the markers that are present, like the protein antigen markers on the surface of cells. Any questions so far? Is this new to anybody? Yeah, does it kind of make sense? Yeah? Okay, cool. So this is what you have on site. And is this the same auto sampler that you have?
Yes.
Yep, okay. And so this one, you can take a tube of cells. It takes about 4 milliliters of fluid, cells in suspension, and put it on here, and it will analyze them for you. Or you can take a 96-well plate and put it in this carousel, and it will automatically analyze all 96 wells or 384 wells. And so you're able to screen with this instrument also. And I guess on site, are you more often doing tubes? Is that what I heard? Oh, okay. And so we were mentioning that this is the original auto sampler. Last year we launched something called the Sidekick Max that can run, where this takes about an hour and 15 minutes or so, hour and 20 to run a 96-well plate. The new one will do it in 22 minutes. So it really improves your throughput. It's not really necessary unless you're screening lots of samples. This instrument's already really fast. But if you start screening, that's something we could look at setting up here. All right. So essentially with flow, our readouts are dots or squiggly lines, right? This is what we look at on a screen. We can look at a squiggly line is a fluorescence histogram that tells us the number of cells over here, the number of cells that are a certain brightness. So to the right are brighter cells, to the left are dimmer cells. And this is like a game of Connect 4 or Plinko or something, where you put cells into buckets, or we call them bins, and the number of cells in each bin, this line will get taller as more cells have a given brightness, right? And so these are our fluorescence histograms. If we combine two fluorescence histograms, one on each axis, then we get a dot plot or a density plot that starts to compare the brightness of one dye against another. So dots and histograms, but there's sometimes when we'll have questions. What does this dot mean? I mean, yes, it's bright in this color, it's bright in that color, but I'm kind of curious about what it looks like. To me, it's just a dot. So just recently, we introduced a version of the Attune that will take a picture of every cell, up to 5,000 images per second or minute, 5,000 per second. It's a wild camera that's in here, 5,000 images per second. So we can look at every dot on your dot plot and say, here's what that cell actually looks like. We can get an image of it. And sometimes that's really valuable because you might have a confusing result here, and you can say with the image, that's confusing because it's not one cell, it's 4 cells that were stuck together. And that's why it's positive for markers that shouldn't be co-positive. It's because I have a lot of cells that it identified as one cell, right? And for very small things, this might be especially interesting for bacteria or exosomes. And so this is the CytPix cytometer. It's the exact same Attune that you have. It's about 3 inches taller so that we can fit this awesome camera inside it that takes pictures of every cell. And this is also the CytKick Max auto sampler that has very large fluid on the outside so it can run a lot faster. So, you know, just ideas for the future that could be things to look at down the road. You're already set up really well, but it's something that we could. You think this would be interesting for your work?
Oh yeah, I think it would be nice.
Yep. Mm-hmm. More.
Well, the other stuff is fine. It's just the camera.
Mhm.
I think it's more for you know, things that you're looking for small population and you want to observe. But overall, I think for most of the things you do in flow, right? I mean, what we have is,
Mhm.
you know.
Mhm. Nice, and it's the kind of thing where if I have a confusing result on the regular tune, I'll go walk across the hall to this instrument and see what's going on.
And what is the probability of things not happening? You know, these numbers. What are some of the culprits?
Oh, it depends on the sample you're running, and we can talk about that definitely. I'll talk about sample prep a bit and how we can get single cell and live cells, healthy cells for analysis. All these things help a lot. And what we find is there are different ways to set a singlets gate, you know, using forward scatter height, forward scatter area, and that identifies single cells and should exclude doublets, triplets, larger things. But what we found with this instrument is you have to make that gate really tight around that population. There's actually a lot of doublets that get into that singlets gate.
Yeah, that's what we do. We do the forward area.
Mm-hmm.
Trying to make that as tight as
Mm-hmm. Nice. Yeah, and we found it, depending on the model, you have to be fairly aggressive with that singlets gate because things will still get into there. Yeah.
So does the camera is only bright field or it does detect the fluorescence as well?
Yep. So in this instrument, it's a bright field camera. And so along with what you get with your you know, fluorescence-based detection, you can say gate on a specific population, gate on my helper T cells, what do they look like? Gate on my macrophages, what do they look like? And so you can see just those. You can also look at all 10,000 images, and if you find one interesting, you highlight it and it populates back onto all of your plots and shows you where it is. So you can see where this interesting thing lives. So and that part's pretty, it opens up a lot of possibilities. So we can definitely chat about that more, but it may be that we can, well, another option. So to get one step more complicated, we've been talking about analyzers, right? Flow cytometers are analyzers. We can also use them to take our cells very quickly, almost immediately, do the analysis, and then ask the instrument to sort cells into different containers depending on what markers they express. And so this is FACS or fluorescence assisted sorting, or various names. But essentially, using flow cytometry, we can tell the instrument to take our sample, and that the calculations here are amazing. How we can analyze our cells, and immediately it knows which tube to send each cell into, so that we can sort out cells expressing a given fluorescent protein, expressing a marker. You could split up your organ on a chip and sort out the individual cells in that organ into different tubes. And so the way that we do that is with the Bigfoot cell sorter. It's about the size of, it's pretty close to the size of that refrigerator, where everything's self-contained. So you just roll it in, the monitor, keyboard stick off to the side, but this can sort 6 different types of samples into tubes, or 8 ways into a 384 well plate, and this takes 10 seconds to put one cell in each of these wells. So another possibility is, if analysis isn't enough, you want to sort your cells. This is a really powerful way to do that also. So any other questions on this so far before we move on? Is sorting something that might have a use here? Yeah. Oh, good. Great. Mm-hmm. Yeah, and Jen is very well equipped over there to run sorts for you. So yeah, that's great. It's a good thing to have in a core where people can come in and run it. So.
And I suppose self-sorting, that would be self-contained, like,
Mhm.
but I think it's not complicated, but doing it every day,
Mhm.
stay there.
Yeah, definitely. Because in the specific, like, you can get the expertise and cell sorting.
Yeah.
User has been doing it over the last 15 something years. That's
Mhm.
why you keep them, takes like a few hours separated to give us, sorted.
Exactly. Yeah.
Yeah, if you purchase, then you have to have somebody expert in that. That's going to be double the cost.
Yep. You need a person to go along with it. Yeah. So we can move on and talk about sample prep for flow, and how just generally strategies to have the best results when you're running flow, to go from samples before you get onto the instrument, and best practices for that. So a few high-level strategies to look at. We can start over here, and one thing we really strive for with flow is to have our cells as live and healthy as possible before we start our experiment. And this is actually the readout of an instrument called the Countess, where you put 10 microliters of your cells into the front of it, push a button, and it gives you the viability and the concentration of your cells in about 5 seconds. So that we know my cells are really live and healthy, and I have a good chance of success in my experiment. If this was 50%, I personally would probably decide to go back and start over and start with fresh cells. That's not always possible, to start over and make your cells healthier. And so the alternative to that is to use a viability die. And these are dyes that we can label our cells with, and it will identify cells that are live and cells that are dead. And so we can ignore all of the dead cells and analyze only the live cells. The reasons for this are, there's multiple. Any antibodies that you're using to label your cells, like I think that's, we're talking about RPE conjugates. Dead cells will non-specifically soak up antibodies. And so they will be positive for all kinds of markers, that aren't actually present, because antibodies non-specifically bind to dead cells. So we want to have as many live cells as we can, but beyond that, we'll use a viability die in every experiment to ignore all of the dead cells in our sample and only analyze live cells. It gives us a much clearer picture of what's going on. Besides that, these are some human natural killer cells that naturally form these clumps. What would happen if I just ran this through a flow cytometer with these enormous clumps of cells? Nasty clogs. Yeah. Exactly, yeah. So the flow cell, especially on the Attune, it's fairly large. It's fairly forgiving. It's not especially easy to clog, but if we have very large clusters of cells like this, it will get stuck in the flow cell, and you essentially abort your experiment, clean the instrument, and then start again. And so we'd like to have our cells in a single cell suspension or something very close to that. 2 or 3 cells stuck together, that's fine, but we don't want long strings of mouse spleen or something going through our instrument, because it clogs it, and it's not single cell analysis. Like we mentioned before with the site picks, it gets confusing if we have many cells stuck together that are expressing given markers. It makes the results confounding of our experiment. And so ways to accomplish this, we can use a pipette tip, aspirate up and down, break up our cells. We can use filters. We use a 70 micron or a 30 micron filter tube and just put our cells in and then run them. Filter out these larger chunks before we run. And then finally, whatever we're labeling our cells with, antibodies, viability dies, proliferation dies, but whatever they are, I work in R&D in our lab developing dies, and we try to optimize them the best we can when they go into the catalog. But for your given experiment, there's probably some more optimization you could do to decide, what concentration of antibody, in the case of antibodies especially, we'd like to titrate them and see how much, as we increase the concentration of our antibody on our cells, our signal goes up to a certain point and our noise increases. And so at what point is this difference between our signal to noise maximized? And this, or ideally here at 250, is the concentration of antibody we'd like to use, which may be a lot lower than how it's packaged in the vial. And so this is an easy first step that may save you some money. Sorry, Mike. But also, it helps improve the clarity of your experiment, right? So anything else I'm missing as far as sample prep that would be valuable for you?
The best way to like tissue count.
Mhm.
What's the best? I know like we one just 6,000.
Sure. Yeah, and it depends on the model. DNase is great because as cells die and the DNA spills out, it just kind of ties all the cells together. Yeah, and you can often see it as kind of looks like shampoo or something in the vial. And so DNase is helpful. EDTA sometimes is helpful.
And then that inhibits the
Yeah, and it can affect other markers you're looking at too. Different enzymes, just depending on the model. There are various different enzymes you can digest your samples with. And we have I think there's a whole web page just for that, how to take your given tissue and here are the different recommended AccuTase, yeah. So yeah, depending on what it is, if it's a spleen, you don't really need anything. You just strain it. If it's a more sturdy tissue, then AccuTase or other enzymes that will actually digest things is helpful. Yeah, any other questions about prepping samples? Okay, we can move on and look at some interesting applications that we can do. But maybe first, should we just go around and just, what would you do with flow if you were to run flow, or what are you doing now? Would you mind if we just share? So I'm talking about the right topics. Would you mind if you were to run flow, what would you do with it? Mhm. Okay. Mhm. Mhm. It's probably antibody based, and then maybe a viability along with that. Okay, great, thanks. Yeah, and how about you? Mhm. Okay. And so maybe a hypoxia dye could be interesting. Okay, great. Yeah, thank you. Got it.
So clinic was basically phenotyping skin cells.
Uh-huh.
You do different preparations skin and as are like melanocyte numbers and
Mm.
in the past I had done immunophenotyping in tumors.
Okay.
So I would give
So lots of colors.
lots of colors.
Yeah. Nice. Okay. So a lot of immunophenotyping.
Yeah. Right now,
Okay. All right. Great. Yeah. Thank you. And for you?
Yeah, ours is mainly phenotyping so
Yeah.
it's
Mhm. Mhm.
also seeing activation antibodies.
Mhm. Mhm. Oh,
So we're using that to power device connection and enhance that. Previously, I did
interesting. Okay.
and currently we have a
All right.
it's a lot.
And how many of those are antibodies?
I think all of them.
Okay.
Yeah, all of them except one is a live.
Okay.
Yeah, that was the only one.
No, great. Nice. Yeah, that makes sense. And then it sounds like there's some stem cells involved in this.
Yeah, and this is
Okay. Yeah, and so definitely you can use a lot of antibodies to identify those. We have some dyes that have, all of our dyes have a protocol with here's how to get the best results. They also have off-label uses.
oh yeah.
And so there's some of our dyes that are meant for other things that will ID stem cells pretty well.
Okay.
So yeah, we can touch on those too.
Yeah, that'd be a good product discussion.
Okay, yeah. And some of those, you can get a similar result with one of those dyes and 9 antibodies.
Oh really? Okay.
To distinguish stem from, non-stem cells.
Gotcha.
So maybe something we could look at.
Yeah, great.
Yeah. Okay, cool. All right. And I heard exosomes.
Yeah,
Yeah.
this is for exosomes mainly.
Mhm. Okay.
Also, we look into the cell
Mhm.
as well. PH1.
This is the autoimmunity.
Yeah, but most.
Okay, great. Yeah. And so, flow cytometry, the site is cell, but we can run lots of things that are not cells, like very small exosomes, beads to capture exosomes. I think that's what you're doing.
Exactly. So yeah, we capture them on 1 to 5 microns. So
Okay.
because they are basically 30 nanometer to 200 nanometer,
Mhm.
similar size of like wires. So it's not
Mhm.
like traditional.
Sure. Yeah. So aggregate a few of them and yeah. Okay. And then how about the two of you? Okay, great. And these are human.
Yeah.
Are you turning fibroblasts into iPSCs? Okay. And then you're turning them back into something else? Cool. Nice. Okay. Yeah. And so this definitely the dissociation conversation could be interesting, but also the same thing with the iPSCs. We can use some non-approved uses of dyes to identify iPSCs also that we could look at. So yeah. And are you both working on the same thing? Yeah. Okay. That's very cool. So, and that definitely has come a ways, that process of making iPSCs and then turning them into muscle organoids. Nice. Do they respond to stimulus? And okay, nice. All right. Well, I prepped a few things we can look at, but then thank you for the whiteboard also, because then lets us expand beyond anything I could have thought of in advance and talk about some of these interesting things. How are you doing on time, Mike? Perfect. Okay, great. So I can run through a few applications. In the case of more basic things, of course, this is how we would do immunophenotyping, where we can take antibodies against given markers. In immunology, they may be CD markers. In muscle or organs, these are probably not CD markers. They may be, I don't know, EpCAM or various cell surface markers, each labeled with a different color of dye. These may excite with the same laser, or they may excite with different lasers. In this case, on your Attune, this is probably going to be on your blue laser. These are probably going to be excited with your yellow laser. And so they each show up in a different channel. We add these to our cells. And then what we get back with our fluorescence histogram, this is what it actually looks like from the Attune, where we'll have what we'd say a peak of cells. Our bright peak, these are cells that express this marker. Our dim peak is cells that either don't express or they have very little of that marker. And so we can say that these are T cells. They express CD3, so these are T cells. We can then take this population, and with our two other antibodies, if those were antibodies against CD4 and CD8, for example, we could identify those T cells that express CD8 and are cytotoxic T cells, and those T cells that express CD4 and are helper T cells. And so, again, as I mentioned, we can take two histograms and turn it into a dot plot. It's essentially a different way of visualizing the data. And so now we can see our cells that express CD4 and our cells that express CD8. And some interesting cells here in the middle that express both. And are these doublets? Maybe we should use that CytPix instrument and go see what the answer is to that. We don't know. Should these be here? Maybe. But this essentially lets us break down a complex population of cells. If it's tumor infiltrating lymphocytes, maybe we have 8 other markers, 12 other markers that really break down what populations are here and how many of each population is there. And so something to mention is a tool called the panel builder. Just go to thermofisher.com/panel builder. It will ask you what instrument you have. So you put in a 4-laser Attune NXT right here, and then it will populate all of the channels that are available here. I think you probably have a V4 here, 4 channels off the violet laser. I think it is. Okay. And so yours would say, V1, 2, 3, 4. It shows you all the channels available. You type in what markers you're after, and it will go through our entire catalog of how many antibodies? 100,000? I don't know. Enormous amount of antibodies that you don't want to go search through. So it will tell you with a dot any antibodies that are available in this combination. And so you just choose, I'd like my viability die to be on the violet laser, which I think is a good choice to have it in the first channel. So that channel's taken. Then my CD105, I'd like to have it on blue. I have 2 choices. It'll tell me what those are and how to acquire them. And then you just complete the rest of the puzzle. You can send this off to your colleagues, send it off to Mike, and he'll give you a discount or whatever you like. But it helps you accomplish getting all of your markers onto this instrument in a way that makes sense. So a tool, you don't have to sign up for this or anything. You just go to the link, it works. So we use this all the time. Also, local expertise. Mike's an expert at this and can give you tips on tweaking this to make it work best for your application. Let's see. Should we talk about compensation briefly? Yeah. So dyes are fairly well behaved, meaning that if you have a channel on your flow cytometer, we try to design a dye so it fits well within that channel and doesn't show up in the other channels on your instrument. But they're not always perfect. So dyes often show up in multiple channels. And what happens is, where our populations should be cleanly in these different quadrants here, what we see is this strange swerving effect, this bending of a population, because the marker that's on this cell is showing up in this channel, but it's also showing up in this channel. And we need to teach the instrument what the individual colors look like so that it can deconvolve them from each other. And so applying what we call spectral compensation or fluorescence compensation, we take a sample of cells or a sample of beads, microspheres, and label a tube of microspheres with each of the dyes in our experiment, feed them through the instrument and teach it what each color looks like. And then when it sees that color again, it knows that it should be just in this channel and it shouldn't appear in this channel. And so often that's one of the first steps in a multicolor flow experiment, is creating your single color controls, running them through the instrument and teaching it what each color looks like. And then we don't have any of this spillover that can be confusing to the outcome of our experiment. So we can use, if we have a lot of cells available, if we're using human blood, we probably have lots of cells. We can just use cells and label them with each of our antibodies. If we don't have a lot of cells available, we can use fake cells. We can use compensation beads that will, if we add our antibody to these beads, they will bind to it, and we can use that to teach our instrument what each color looks like. Right? Is this something that you use now? So there's the Ultra Comp, the eBeads Plus is the newest one. The ABC beads is another option. I don't know, any guidance on which one to use?
I would say that there's a difference between the plus and the polymer. They tweaked it so that the polymer dyes, when they come to the environment, you don't have so bringing violet, super brights.
Yeah, thanks. Get rid of some of those artifacts that the new polymer dyes have introduced. So as instruments evolve, the dyes evolve, and these have to evolve also to keep up with things. So I think we can talk briefly about viability, because I think this is really, again, one of the most important parts of a successful experiment. And when we're talking about cell viability, we can take various different dyes, add them to our cells in the tube, and our viability dye. There are different ways of accomplishing this, but either way, the readout is the same, where a viability dye will enter a dead cell and make it appear very bright. They will be excluded from a live cell, and so a live cell will be dim. And so on a histogram, we can see our bright cells, our dead cells. The dim peak here is our live cells. And so if we're doing like a T-cell killing experiment, maybe we're interested in the dead cells, but usually we're not. Usually we just gate on our live cells and then ignore all of our dead cells. If we're over at the university in the BSL-3 facility, we probably have to formaldehyde fix our samples before we go analyze them. And not all viability dyes can survive that formaldehyde fixation, but some can. With propydium iodide, which is a very old viability dye, if you fix your cells with formaldehyde to preserve them or to kill off any viruses present, all your cells are dead, and so they're all bright, right? It's not so helpful in that case. So dyes called live dead fixable viability dyes, where this stains DNA, these stain protein in your cells covalently, and they're permanently attached to your cells. Even if you fix or put them in the refrigerator for a month, the dye is still on your cells. So we can fix our cells and still distinguish live and dead. And this is something we've spent some time on in the last year, refreshing these. So now there are 14 colors available. So again, we talked about the panel builder tool. Now, no matter what colors you're using on your experiment, there's one of these dyes that will fit. You can add it to your experiment. So it's easy to find an available open panel and slot one of these dyes into it. And some pointers on these. Again, as I mentioned, everything has its protocol, but then there's also the secret off-label protocol that I can tell you about for how to use them. Generally, with these dyes, they show up in your freezer. You don't have to keep them in your freezer. It's recommended to, but they're stable. You can take them out, dissolve them in a solvent that's provided with the dye in the kit. Add 1 microliter to your cells, incubate, and in the protocol, this says 20 minutes. You can do it as short as 1 or 2 minutes, and it's adequate. It's very fast. So if your cells are sensitive, yeah, I did this testing last summer, and 2 minutes is normally enough. If you want really bright dead cells, go the full 20 minutes, but it's really almost an immediate labeling step. So label, and then in the protocol, it says, we'd like you to wash your cells. Then you could label with antibodies on surface markers. Then you can fix and permeabilize your cells, poke holes in your cells, and then label with antibodies against targets inside your cells. All of these things are optional. We don't even have to wash these dyes back out. You can just label and analyze. And this is something I tested a lot last summer also, that it's adequate just to add the dye and go run on the instrument. Ideally, you'd wash it back out. I'd still recommend it, but you don't have to. This labeling step where you add dye to your cells, because these dyes label protein, it becomes challenging if there's protein present in your media that your cells are in, because the dye will just label the BSA that's in the media with your cells. So generally 1% protein in with your cells is okay. 2% or higher will change the behavior of these dyes. It's true, yeah. And so some of those things, if you're going to do something different, just try it and see. Some of them, there is some flexibility there where you can use 1/10 of the dye and it still works okay, but not for all of them. So yeah, if you want to do something interesting, give it a shot. But generally, the protocol is kind of designed to give you the best results. It's in there. So let's see. I included a few other things here, but we may veer off course and talk about some of the things that you had suggested. We included viability, but oh yeah. So with your viability dyes, these figures where you have, we have really dead cells and nice live cells, and this makes it look like, oh, this is going to be a piece of cake to gate on my live cells because they're so far separated from my dead cells. It's obvious which one is which. Do you think this happens in the lab? No. Yeah, so I really killed these cells to make them that positive. And these were nice 100% live cells mixed together, and it makes a nice figure. But in reality, especially if you're using blood, where you have lymphocytes, monocytes, neutrophils, different sizes of cells, cells from different sources, cells from different tissues mixed together, you might have something more like this, where you have 3 peaks. And I get this question all the time. I don't have 2 peaks, I have 3 or 4. Where should I gate? And so in this case, this is a cell line. These are T cells, but with 3 peaks. And what we can do is gate on the peaks and see what they are. We can gate on this one that I think is the live cells, And by scatter, we have high forward scatter and low side scatter. It's a good sign. But then we can go to our CytPix instrument and say, what do they look like? Well, they're nice and round, and they're smooth. They look intact. I think those are live. This other peak that's really bright, higher side scatter, lower forward scatter, I think those are dead. With the CytPix, we can see the magnification is a little off on this display, but they're bumpy. They're not so round. There's a lot of texture in the middle. I think those are dead. But what is this third peak? And what it turns out is these are so far dead that the viability die has started to leak out and they've become dimmer. So these are dead, they're just really dead. And so that dye that labels protein, there's just less protein here now because they're falling apart.
So the second row is apoptosis, third is necrosis.
Potentially, yeah, depending on the treatment. It could be. Yeah, so in this case, if you want live cells, it's easy. But you know, either relying on scatter, knowing that forward and side scatter change along with viability, or looking at the image, it really helps you clarify. And also multiplexing with antibodies. If you have lymphocytes and monocytes, that's often why you see multiple peaks also, because monocytes are much larger than lymphocytes. They will be brighter on this. So in whole blood, I often see 4 peaks, and it's totally normal. It just depends on what you're studying, right? Have you seen this also?
Yeah. I've never seen the really bad structural degradation. That's blue.
Yeah. And they do need to be really dead to see that. It's not all that common to see them that far gone, but yeah, it's another source of what's happening.